Howdy, this is Browdy. So I'm coming at you with the second part of the genetics unit. Now, I think based on the way things have been broken up between class, I'm actually going to have one last section. So the second lecture, I'm going to kind of break up into two videos because there's one part that I want to save for a little bit later. But for the sake of time, I'm just going to get right to it. So in the last video, we talked about Mendel, who he was, what he did, and just heredity and genetics in general. And so this time around, now that we know some of the vocab, so genotype, phenotype, dominant, recessive, homozygous, heterozygous, we can talk about Punnett squares and how we can predict traits based on probability. And so, first and foremost, a Punnett square is basically this box that we see right here. Now you'll notice that I've used P's in the past, but they use F's here, and I think it's good that we have two different versions because you can actually use whatever letter you want to represent anything. I think the most important thing is making sure the capital and lowercase versions are easy to distinguish. But here, I've used P in the past for purple versus white, but they use F for flower, and that's fine. And so what this is, first and foremost, is that the letters on the axes or on the outsides of the boxes represent the genotypes of the parents. And so this cross is a big F, or big P, homozygous dominant parent, meaning they're a purple flower, crossed with a little f, little p, a homozygous recessive parent, so a white flower. So this is what Mendel did. This is his P generation. Purebred purple crossed with purebred white. Now the boxes on the inside show the possible genotypes of the offspring. So all these in here are showing, well, there's 4 boxes, so that means there's potentially 4 possible combinations of alleles. Now we realize here that that's not actually the case in this specific cross because this parent can only give big F, this parent can only give little f, so we've got all of the offspring being big F, little f. What a Punnett square actually does is it predicts the ratio of genotypes and phenotypes in the offspring. And so here, because this parent can only give a big F and this parent can only give a little f, all of the offspring are big F, little f. Now the way this works is that you see, okay, I've got an F here, so I carry this F into this box and this box. And same thing here. Now I carry the little f's over here into all the boxes to the side of it. And same thing here. So this box has a big F and a little f. This box has a big F and a little f. This box has a big F and a little f, and so on, right? Now, this is just another example. If green is the dominant color for pea pod plants and yellow is the recessive color, these genotypes correspond to phenotypes. So big G would be green because it's homozygous dominant. Heterozygous, big G would also be green because green is dominant. And little g would be yellow because that's recessive. And the only way to show the recessive phenotype is if it's a homozygous recessive genotype. Now, the first cross that we see, actually no, sorry, this is not on the notes. This is one more until the notes. This cross that we see here is this second cross that Mendel would have done. So this parental generation were 2 purebred plants, and those would give you heterozygous offspring. Here he's crossed the heterozygous offspring to give you the F2 generation. Now what I want to show you here is actually the ratios. So here he crossed a heterozygous purple plant with a heterozygous purple plant. This big F gets carried to here, this little f gets carried into these. This big F gets carried into here, and this little f gets carried into these. So in this box, you've got big F. In this box, you've got big F, little f. In this box, you've got big F, little f. And in this box, you've got little f. So what we see here is a ratio of the genotypes. So we have 1 big F to 2 big F, little f's to 1 little f. And so these colons we say as 2, so 1 to 2 to 1. And that's a ratio. It's essentially saying that if they had a number of different offspring, maybe they had 8 instead of 4. but the way it would work out is you'd have twice as many of these as you would of these and these. Now the phenotypes don't work out quite the same way, because remember, both this and this genotype give you the purple phenotype. So there's actually 3 purple for every 1 white. And so we'd write that as 3 purple to 1 white. Now here's the practice that goes with your notes. So here's the first one, and you should see this here on your notes on this side. So the parental genotypes of this cross, this is the P generation, or creating the F1. So the first cross is homozygous purple with homozygous white. Now I've gone back to using P's here, and being able to switch between letters is a really important skill. But so this cross has a homozygous purple parent, big P, with a homozygous white parent, little p. And so we write that as big P by little p. That X, that multiplication symbol, says that they're crossing. Now, just like we saw in the first one, the ratio here is going to be 4 big P to nothing, because there's nothing else, right? You could just write 100% big P, or you could say all big P in this case. They're all heterozygous for that genotypic ratio. The phenotypic ratio, since it's just purple and white, we can say there's 4 purple to 0 white, because all of these would be purple. You could just say all purple as well, and that's okay. Now this next cross down here on the notes, we're crossing the F1 to get the F2. So here we have the heterozygous purple plant crossed with the heterozygous purple plant. They're both big P, little p, and you'll see that over here. When that happens, you carry the big P here, the little p here, the big P and the little p, and you'll get 1 big P, 2 big P, and 1 little p. And so we would have that ratio as 1 big P to 2 big P to 1 little p. You could write it this way, or you could write it as I did here in the notes, and either one is okay. But essentially that means that you'll have the same amount of homozygous dominant and homozygous recessive no matter how many offspring you have, and there will be twice as many heterozygous offspring. The phenotypic ratio is 3 purple here to 1 white. So no matter how many I have, I'll have 3 times as many purple offspring as I would white. Now there's also the chance that we cross a heterozygous purple parent with a homozygous white parent. So heterozygous with homozygous recessive. And we'd set up that cross like this. It'd be big P by little p. So big P, little p. And when you carry that out, the big P would go to these, the little p would go to these, and then this parent would contribute a little p to every box. And what you end up with is 2 heterozygous offspring and 2 homozygous recessive offspring. So the genotypic ratio would be 2 of big P to 2 little p. But since it's 2 to 2, we can actually reduce that to just 1 to 1. So the genotypic ratio is 1 big P to 1 little p. Essentially meaning that for every heterozygous offspring you'll have, you should have 1 homozygous recessive offspring, meaning you'll have the same number of each, half and half. The phenotypic ratio is the same, it's 1 to 1. For every 1 purple kid you'd have 1 white offspring. And that's the Punnett square practice. But there's one last concept to talk about, and this is the idea of a test cross. And so a test cross is a way that we can determine if the genotype of an individual with the dominant phenotype is either homozygous dominant or heterozygous. So if, say, Mr. Browdy had a pea plant garden, you wouldn't put it past me, right? Say a random purple flower pops up, and I say, cool, but I want to know, is this homozygous dominant or is it heterozygous? Because either one of those genotypes could give me the purple phenotype. What I do in that scenario is I cross it with a recessive white flower. The reason being, so we'd write that cross like this, actually, before I say why. we'd say big P dash crossed with little p, because we don't know if that's big P, or if it's big P. If the plant were big P, and it was crossed with little p, well, we've seen that cross before. That would give us all heterozygous offspring, so all purple. And if all the offspring were purple, that would tell me, okay, that was homozygous dominant. But if some of the offspring are purple and some of the offspring are white, meaning half and half, that would tell me that this was heterozygous, because the only way any of the offspring could be white is if they got a white allele from both parents. For that to happen, my purple flower would have to be heterozygous, meaning it has one purple and one white allele. And that's what we have down here, and we call that a test cross. Now, the very last little bit on the notes is this big square down here, and I'm going to leave that out for the time being. And so we might see a third video lecture, and that's all good. I just kind of wanted to keep this consistent with the way we've done it in class, and I'll kind of make that one last topic. So thank you for hanging in there with me. I hope this has been helpful, and I hope it's been informative. I tried to keep it quick, but of course, if you have any questions, just let me know. Browdy is outie, and I'll see you in the next one. Take care.